Okay, good day everyone. Today we're gonna discuss the microscope practice set number one. If you need the lecture, kindly click the link below for you to access the lecture part. So it will be redirecting you in our Google Sheet. Okay. So let's try now to discuss number one. Which of these describe the correct use of microscope? So for number one, the answer here is letter So letter B. When focusing in HPO, slowly adjust the LPO using the coarse adjustment knob first, then the fine adjustment knob. Okay, so again, LPO is for coarse adjustment and the HPO is for fine adjustment. Now, how about letter A? When focusing in HPO, adjust the fine adjustment knob. No, so this one is really wrong. Okay. Next, how about letter C? Start with the objective with the highest magnification. No, you always start first with lowest magnification, unless you are familiar with the uh, with the uh, with the cell that you're gonna view. How about letter D? When viewing with HPO, use the coarse adjustment now. It's a never thing to do that. Why? You're gonna break the glass and it can penalize you with breakage of the glass as well as the destruction of the specimen. So letter D, it's a no when viewing with HPO. You only have to do the fine adjustment knob. Okay, so next, number two, which of this type of microscope you will use if you want to look at the inner layers of the mitochondrial membrane? Of course, if you want to look at the details of the organelles, you will use the transmission electron microscope. So it uses electron waves rather than the light. Okay. So compound light microscope, you cannot see the details out of it. The fluorescent microscope is used for examining tissues as well as for interference light microscope. In fact, if you want to view a live detailed of the cells, it is better to use sometimes the interference light microscope. Next, how about number three? Which of these is false about using electron microscope? Letter A, electron microscope need a large space and is very expensive. Yes, of course. That's why we don't use that usually in school. Next, how about letter B? Electron microscope cannot visualize live cells in details and movement. Yes, you cannot do that because it is usually dead. No, the cells and the organism usually dead because it uses a vacuum and heavy metals for staining. How about letter C? Electron microscope can visualize detailed organelles. Yes, this one is correct. Okay. How about letter D? Electron microscope uses property of lights. No, we don't use properties of lights for electron microscope because we simply use electron waves. So we cannot do that. Okay. So next, number four, the image of the cell. Why this one is called? The image of the cell is 4.8 millimeter and the actual size is 32 micrometer wide. What is the magnification? Remember the formula for magnification. That is I over O. That right, image over the actual size of the image. So image over the actual size. Okay. Now, uh we can simply use this formula directly. The image of the cell is 4.8 millimeter and 32 micrometers. So we cannot directly use that, right? But we all know from the previous discussion that 1 millimeter is equals to 1,000 micrometer. We can simply memorize this part. But for you to imagine that the millimeter of a ruler, imagine 1 millimeter of the ruler, divide that by 1,000 times, that is the 1,000, that is the 1 micrometer. And so 32 micrometer wide, so 32 micrometer, okay? So, okay. So 4.8 millimeter, convert it into micrometer times. I'm gonna put the 1 millimeter at the bottom part because I want to cancel this out, and then the 1,000 micrometer on the upper part. And so this one is 4.8 times 1,000. Remember your math before, you're gonna add three zeros out of it. One, right? So we have one, two, three, right? So that would be 4,800 micrometer. And now we are ready to use the formula. We have A of image over the actual, and that is 4,800 micrometer over 32 micrometer. And so how we're going to solve for this one? Now remember, if doing this, that could be 48 times 100 over 32. This will going to help simplify our math. So 48 and 32. So 48 divided by 4, it's 12. 32 divided by 4 is 8. So that would be 12 over 8 times 100. And 12 divided by 4, it's 3. 8 divided by 4, it's 2. So that's 3 over 2 times 100. And what is 3 over 2? That is 1.5 times 100. And 1.5 times 100, adjust it with 2 decimal places. So that would be 150 times. So for letter B, our answer is 150 times. Okay, now how about number 5? Let's look at number 5. A starch grain is viewed with 200 millimeter. The magnification is 800 times. What is the actual size of the image in micrometer? Let me just change the color of my ball pen. So this time I'll use blue. Okay. So take note, we have M equals to magnification equals to A image over the actual, right? And so we are looking for the actual size, derive it algebraically. That is just image over the magnification. So the problem here is that the starch grain is viewed 20 millimeter. So that would be the image, right? So this one is the image. But our choices here is micrometer, right? And so we need to convert that again, 20 millimeter times 1 millimeter and then 1,000 micrometer. Cancel out, and that would be 20,000 micrometer. And so we are now applying the formula that is 20,000 micrometer over 800 times. So now if we're going to look how to simplify this is 800. The two zeros will be canceled. We are left with 200 over 8. 200 divided by 100 is, uh, sorry, 200 divided by 2 is 100. 8 divided by 2 is 4. And then 50 over 2, and that would be 25. Okay, so the answer here is letter B, 25 micrometer. Okay, and that ends our discussion for the microscopy. Thank you very much for listening.